we had overlooked a little bit of the processing, the mechanical processing at the mouth. So I want to just add a quick word about saliva, because we're taking a bit of a blended approach to the mechanical and chemical digestion of food. But you know that the bolus of food, mushed up food mixed with saliva, moves through the esophagus according to the peristaltic contractions of the esophagus. And that can be initiated voluntarily, but the pharyngeal and the esophageal phases are involuntary. Once you start to swallow, you can't stop. But a word briefly on digestion in the oral cavity. There is some that occurs as a result of the secretion of saliva. And you don't have this slide, and you can be mad at me now, but I'll include it later, or you can write it down. So saliva lubricates the fluid and allows it to mix, making a bolus. And you should be aware of this from lab 2 weeks ago. Some of these points you'll be aware of from lab last week, but certainly as far back as 2 weeks ago. Saliva is largely water, but has a few other ions and special things in it. It's produced from the interstitial fluid that is made from blood supply. It has some salts, it has mucus, and enzymes that break down carbohydrate. Matt probably told you about that saltine cracker example, where as a kid, if you just chewed it and let it sit, eventually it would start to become sweet. And that's the result of specific enzymes in saliva that break down carbohydrate. Now that enzyme in particular is amylase. And a little cheat sheet for any enzymes is usually they end with ASE. They end with A's on the end. Amylase, lipase, protease. And that prefix will tell you what they break down or what they cleave. So it has some enzymes. It even has a lipase enzyme that breaks down fat, but it's not currently active in saliva in the mouth. It only is activated when it reaches the stomach at the low pH of the stomach. Still not a big contributor to fat breakdown. We'll see the enzymes that are the major contributors to fat breakdown today. It also contains lysozyme, which is an antibacterial agent, so it helps to break down food. It mixes and lubricates the food, and it protects the surfaces within the mouth, the teeth, gums, the esophagus, and the stomach. Otherwise, we would have completely overlooked saliva, and I think that's a bit of a disservice. So, some main points about saliva. Take it for granted. It's also really interesting. I just personally find this cool from a sport physiology and ex-phys point of view. There are receptors in the mouth for sugar. Now, it's not immediately clear what that means. Some glucose gets absorbed through the mucosa of the mouth, not a whole lot, but there are signals sent from the mouth to the brain that say, energy's incoming. And from a sport physiology point of view, this is really cool because you can swish sugary fluid around in your mouth and then spit it out. And you get a performance benefit from that. The signal is still sent to the brain saying that energy's coming in, and even if you spit that fluid out, you can see some performance improvements if that's what you're measuring. So really interesting. There are sensors in the mouth for sugar that relay signals to the brain, help turn the lights back on, and improve sport performance. So a neat little tangent on the idea of kinetics, sport performance, and sport physiology. Now, leaving this behind, food is mixed, the bolus is swallowed and it enters the stomach. So the lower esophageal sphincter opens, allowing food in, closes again to prevent reflux. We are in the stomach, and you know the general shape of the stomach. There's 4 main areas. Cardia at the top, the fundus, this upper bulge on the left-hand side. The body, by far the largest part of the stomach. And then the pyloric area, which can be divided into subsections, but we'll just call it the pyloric area. And it's so named because the distal end is where the pyloric sphincter is located. And this is the sphincter, the round muscle that allows the passage of food from the stomach into the small intestine. And this should be reviewed. You should know this. It kind of looks like an inflated sausage. And it is. It's one of the most extensible organs or parts of the digestive system. Light J-shaped, extensible sausage with 2 obvious curves, the lesser and greater curvatures, which we've seen referred to in our section on blood supply way back in the 2nd series of slides, if you remember way back that far. We'll bring a few of those points back today as we talk about blood supply to the pancreas, the stomach, the liver. So the orientation of the stomach shown here. Notice also the multiple orientations of muscle fibers. So you see there are 3 layers peeled back with the fibers all in different orientations. Longitudinal, in line with the stomach, circular, around in bands around the stomach, and oblique at an angle to both of those layers. Multiple orientations, which allow multiple vectors of contraction and really effective ability to mix and churn and push and compress food in the stomach. I suppose at this point in our discussion, I should say the bolus in the stomach, but that's soon to also change. So layers of smooth muscle, allowing churning, mixing, and then placing the bolus in the stomach to be processed a bit more. And you'll see visibly these rugae or folds inside the stomach that allow for its distension, that allow it to expand and stretch and accommodate those large buffets that you might have on the odd weekend. There's no Mandarin around here, but if you've ever been to a Mandarin, you know, those kind of buffets where your stomach just feels like a brick afterwards. The rugae expand and allow the extension of the stomach. And you can see these very visibly looking inside the stomach on the right-hand side of the top. You can see the folds visibly. These expand to accept any incoming food, at which point the stomach starts its job, which is mainly of storage. The stomach's main job is mainly to store the food and meter its delivery into the small intestine. So it expands to accommodate and stores that food, mixing with some things and contracting, but mainly storing until the small intestine is ready for it. So that mixing is again peristalsis, rhythmic churning of food in the stomach. We'll talk more about this on the next slide, but that rhythmic churning has a purpose: to mix the incoming bolus with the gastric juice that the stomach secretes. And you know some of the components of the gastric juice already after doing the chemical aspects of digestion lab last week. You know the major components. And at this point, after the stomach mixes the bolus with the juice, it becomes chyme. We call it chyme. Very similar, although more liquid than the bolus of food that left the mouth through the esophagus and entered the stomach initially. So the food is much more liquid when it's in its form as chyme, and that allows a lot more propulsion. Now that propulsion is due to the contraction of those 3 layers of muscle tissue. And largely, the stomach is trying to push that chyme towards the pyloric sphincter so it can leave the stomach. But it's actually very hard to leave the stomach. Pyloric sphincter is not very open, and it doesn't open very much when it's open. There's only a small opening that allows maybe 3 to 5 milliliters of chyme to get through at any given point in time. And so anything that doesn't leave the stomach recirculates. It's pressed against the walls and returns into the body. And the circuit continues. Mixing with gastric juice. If there's any other solid food bolus that I forgot to mention would be stored over in the fundus on the left-hand side, that mixes with chyme, and gradually the liquid juice is expelled from the stomach. But not all of it goes out at once. That process is called gastric emptying. And in the sports drink field, which is where I did a lot of my training and where a lot of my research lies, we would measure gastric emptying to see if different formulations worked better. Higher sugar or maybe some protein would help sports drinks work better. You know how you measure gastric emptying in that field? You know what you put into the stomach. You have to go in and get whatever's left and compare the two. And the difference is what would have been emptied from the stomach. So if you're an unfortunate person to be in one of those studies, you drink a beverage, your stomach is aspirated and everything is sucked back out again, and the two are compared. And whatever's not there was emptied from the stomach. Luckily that work's mostly been done now, so you don't need to volunteer for a study like that. I can tell you what kind of sports drink is optimal for helping gastric emptying. Eventually, all the food will leave the stomach in a matter of 2 to 4 hours, but until that happens, it's mixing, churning, and then brief, small expulsions of chyme into the small intestine. So to outline the functions that we've described, what does the stomach do? It takes the bolus, which is food and saliva, and then mixes it with gastric juice to form chyme. Much more liquidy. Food particles are suspended with easy access from enzymes to help break them down. Largely, the stomach serves as a storage reservoir. It mixes and churns and tries to expel, but it's not very successful. It's a slow process. For this mixing to occur, and so that chyme can be formed, of course, the stomach needs to secrete gastric juice, whatever that is. Another big function of the stomach, to secrete gastric juice. It's mainly acidic, containing hydrochloric acid, with some other enzymes that you saw last week. Pepsinogen, that is converted to pepsin, that can help to start breaking down proteins. Intrinsic factor, which isn't immediately clear, but it helps with vitamin B12 absorption. And lipase, again, ASE, an enzyme that works on lipids. not the major lipid breakdown enzyme in the body, but one of them. Another function in the control of digestion is that some cells in the stomach secrete gastrin, which is a local, you can think of it as a hormone, a local signal that sends a message to other cells in the stomach whenever food is present. And we'll go over that process in a bit of detail in the next two slides. So gastrin is a local signal, hormonal factor, that signals other cells in the stomach to do their job or to ramp up the work they're doing. What's not included here, that's a tangent that I don't care if you remember, that I think is really interesting, is there are also a lot of hormones secreted that influence appetite at this level. You heard of ghrelin or PYY? These are appetite hormones. And there's a lot of research, given our current obesity epidemic, on how to maybe release these in without the presence of food, or make those feelings persist longer so you don't want to ingest more and more food. But a lot of hormones are also released here that we're not going to study in depth. Let's focus instead on gastric juice and what gastrin does to help produce gastric juice. Gastric juice, synthesized, excreted, expelled from gastric pits. A lot like sweat glands in the skin, if you think about it. There are individual glands within these pits that produce the primary fluid, if you will, and that is expelled into the lumen of the stomach as the pit fills up. And so you can see the pit roughly tooth-shaped here with the roots. The pit is what collects the excretions from the individual cells or the individual glands in the stomach and then releases the mixed fluid into the lumen. And the cells that contribute to the gastric juice are shown over here on the right-hand side. Now, you've talked about chief cells a fair bit. This is the cell that secretes pepsinogen, which is the precursor to that protein enzyme that you saw in lab last week. It also secretes the enzyme to break down fat that's not very active at this point. The chief cells responsible for those enzymes. Also of importance are the parietal cells, which make the environment acidic, which make the juice acidic. It's the action of these cells that largely produces hydrochloric acid. That's not to say the other cells aren't important, but those are noteworthy and things that you've probably heard mentioned before, if not seen. But those two cells, with the mucus that is produced by the surface and the neck cells, is what makes gastric juice. It's a combination of mucus, the hydrochloric acid from parietal cells, and the enzymes from the chief cells. That is what the gastric juice is made of. Often you'll ask the question, why don't the cells of the stomach themselves denature or unravel or are digested? And that's the role of these mucus-secreting cells primarily, to help coat and protect the layer of the stomach so that it is not damaged by the gastric juice and the chyme as it mixes. What we haven't talked about yet, the only other type of cell on the slide, are G cells down at the bottom, which are responsible for secreting gastrin. Somehow, these are connected to the other cells that make gastric juice. somehow this signal, gastrin, influences these other cells, and we'll see that coming up. But they're not directly involved in synthesis of the fluid. They're involved in sending the signal to synthesize the fluid. So how, for instance, do we make the gastric juice acidic? And this will end up being a fairly busy slide, but we'll approach it in stages. So we want to focus on hydrochloric acid in the gastric juice, so we're looking at a parietal cell. That's the cell that makes or secretes hydrochloric acid. And you can see the initial precursor, the first step in this process, is that in response to some signal, these cells will actively pump out hydrogen ions. Now, actively is a special word that implies energy requiring. This is an active process that requires energy to pump out protons in exchange for potassium ions. So energy, you can see, is the breakdown of ATP in order to do work. The exchange of ions occurs, and there are more protons in the lumen on the left-hand side, as you look at the screen, than within the cell. Now that wouldn't do. To make hydrochloric acid, we need chloride as well. The potassium that was just brought in by that energy requiring process leaks back out. There are pores in the parietal cell, a lot like we had potassium leak channels in cardiac myocytes. There are potassium leak channels in the parietal cells. And so this chloride escapes. All the work that you did, the energy you used to pump that in exchange for a proton, is undone. But this is an important step in the process, because if you're keeping track and tallying the charge in the lumen and inside the cell, we now have a proton in the lumen and a potassium ion in the lumen, two positive charges. So something has to happen to equalize that charge, and that something is shown in the second purple circle. Chloride travels out. It's drawn by the positive charge into the lumen, and then bam, makes hydrochloric acid. So we exchange a proton for potassium. The proton combines with chloride that's drawn by the potassium after it leaks back out, and that's where the acidity comes from. But interestingly, there's not just an endless supply of protons. We need a source of these to pump out in order to make hydrochloric acid. And oddly enough, those come from our old friend carbonic anhydrase. The same reaction that we saw in the blood that helped deal with excess PCO2 and helped CO2 be transported through the blood. Here is a supply of hydrogen ions for the production of hydrochloric acid. In the same way that the equation wants to be balanced in the blood, it's also balanced here. We remove protons in this process, and that pulls the equation to the right. If we just got rid of those protons, we'd be left with bicarbonate building up. That bicarbonate leaves via the blood in exchange for chloride, again to keep the charge balanced. And that's where our source of chloride is for the production hydrochloric acid. But what sets this series of events in motion is the question. It doesn't happen automatically. It can happen in response to stretch to some degree, so physical stimulus. But the main stimuli that induce parietal cells to make hydrochloric acid and start to secrete gastric juice are acetylcholine from the sympathetic nervous system. Enteric nervous system. And our friend gastrin, which we identified on the last slide. When gastrin is produced, it travels a very short distance in the blood, binds to a parietal cell, and makes it start to produce more hydrochloric acid. This should be ENS, by the way, enteric nervous system. So production of hydrochloric acid and magnification or initiation of that process by gastrin secreted from the G cells. So what does that do to the food in the stomach? We need gastric juice to be acidic. We need enzymes to be secreted from the chief cells. What does that do to the food then in the stomach? So hydrochloric acid, as the food bolus mixes and becomes chyme, helps to unravel proteins, unravel and denature proteins within that food. The acidic environment unravels these proteins, and then through the action of enzymes that are also secreted, we can now start to break them down. We saw that protein digestion, for instance, started in the stomach last week. If all the conditions are present, pepsin, which is activated by the low pH, will cleave that protein as it unravels. And really the same thing would happen with any enzyme if the conditions were favorable and it was in contact with the thing it was supposed to break down. Here, pepsin's in an ideal environment. Pepsin is the appropriately shaped scissor to cut this yarn. And so proteins will start to be degraded. But not all enzymes will cleave all proteins. Or not all lipases will cleave all fats. Not all proteases will cleave all proteins. The scissors have to be the right shape for the thread in this case. Imagine if your scissors were too small, or imagine if they were those child safety scissors, or you were trying to cut a giant rope. There are appropriate matching scissor and thread combos. So we have the action of pepsin working here. The lipase should also work, but it only provides limited cleavage of fats from triglycerides. By far the majority of free fatty acid cleavage comes from pancreatic lipase, which is coming up. If I'm not mistaken, that was the example that you had in lab anyways. Pancreatic lipase does most of the heavy lifting as far as fat breakdown is concerned. And so while we have some digestion occurring, there isn't much, and therefore not much absorption occurs other than those solutes that are already dissolved or freely moving. There's some there's some absorption of elements that are suspended in liquid, but very few macronutrients are absorbed in the stomach. There's some carbohydrate absorption, very little protein and fat absorption. It's much more ions, alcohol, or water itself. So as we mentioned before, typically the stomach will empty, depending on the bolus, in 2 to 4 hours. But the type of food and the composition will affect how that emptying proceeds. If it's a very loose liquid fluid with simple sugars, it's absorbed quickly. It passes through the stomach quickly. High carbohydrate meals tend to move out faster than other mixtures. A protein-laden meal takes a moderate amount of time, and then it usually takes the longest for high-fat meals to be processed and leave the stomach. Notice this is just gastric emptying, how long it takes for the stomach to empty. These aren't feelings of fullness. That's a completely other topic that you'll probably encounter in nutrition. They aren't one-to-one. So fast emptying with carbohydrates, which is really good if you're trying to perform at a high level over a long term in a marathon. You want really simple sugars. They empty quickly to provide fuel to the body. You don't want heavy proteins and fats. So how then is this digestion regulated? It's worth mentioning now the phases of digestion at the stomach in preparation for our description and traveling into the intestines. These processes don't always occur. They're not constitutive and always active. The phases of digestion occur in 3 steps. The first is the cephalic phase. Remember, cephalic was a prefix or a name describing of the head. Cephalic is a feed-forward mechanism that stimulates gastric secretions, stimulates motility, stimulates the production of saliva in anticipation of a meal. The cephalic phase describes the process of your mouth watering as you look at that piping hot pizza on the table in front of you, or whatever your guilty pleasure is. The cephalic phase leads to the gastric phase, and the gastric phase emphasizes mixing and breakdown and absorption, but only if food ends up being ingested. If food isn't ingested, there's no feedback signals and the cephalic phase is overridden. Your mouth won't water forever. The gastric phase, what we'll explore next, describes when food is in contact with the stomach, what are the neural and hormonal signals that bolster that response. And then similarly, as food passes through the intestines, it reinforces the signals that help to secrete gastric secretions and enzymes to help with absorption. So it's a stepwise process. And the gastric and intestinal phases feed back and reinforce those signals. So since we just spent some time talking about gastric juice, the regulation of gastric pH, and how gastric motility occurs, it makes sense to look at the gastric phase of digestion. Your mouth isn't always watering. Your stomach isn't always contracting and churning and mixing food. You aren't always producing high amounts of gastric juice. The initial stimulus comes from food entering the stomach. Food enters the stomach. There are receptors in the wall of the stomach that detect changes in pH. Food isn't an acidic pH, and so it will affect the pH within the lumen. And the stomach, which is shriveled initially, will open up, will be stretched. These signals are evidence that food is present, and you need to do something about it in order to process and digest it. Those sensors relay through the enteric and parasympathetic nervous systems signals to secrete gastric juice and increase motility. These signals also initiate the release of gastrin. Gastrin, the enteric, the parasympathetic nervous systems start to produce gastric juice. Gastric juice mixes with the food. The stomach, through peristaltic contractions, churns and pushes the food towards the pyloric sphincter. Maybe some escapes. If not, it still mixes and churns. And as long as that pH and stretch are maintained, this process continues. Maybe you disrupt pH more and disrupt stretch more by continuing to eat, in which case these signals become stronger. But eventually, as the chyme is expelled from the stomach, pH returns to normal, stretch returns to normal, and these signals get weaker. They abate. These signals go away. And so the churning, the peristalsis, production of gastric juice, all decrease back down to a baseline low level. Notice, if the cephalic phase is engaged and you're anticipating that food is coming in, you might kickstart some of these responses, but if there's no distension and if there's no change in pH, those signals aren't reinforced and they shut back down right away. So this is feedback confirming that food has entered the stomach. So, questions about this? So far, so good? You good? All right. A few minutes left. We can approach the small intestine with a primer on the pancreas and liver that help to prepare that foodstuff as it enters the small intestine. So the duodenum being the first branch or the first section of the small intestine is where food is expelled to next. The chyme is expelled into the duodenum immediately, where it mixes with secretions from the liver and the pancreas. Remember, acidic environment, some enzymes need to be neutralized. pH needs to be neutralized, which is largely the function of the pancreas. So you know the pancreas well from your studies in lab. Here you see the pancreas and the duodenum. I've tried to draw a little red line here to show you the division between the two. This is the inside of the small intestine, and you can see some of the ducts that relay enzymes and alkaline enzyme solution and bile from the liver into the duodenum proper. So you know it lies posterior to the stomach. This is the site where we get most of the enzymes that break down carbohydrates, proteins, and fat. By far the major source of these enzymes, and they're what we would consider broad spectrum enzymes. They work on many different types of bonds. They're versatile. That's not to say that a protease can also cleave a fat molecule. A protease will only work on proteins, but it will work on many kinds of protein bonds. Think of it that way. So we have many wide spectrum enzymes that are released from the pancreas. Also sodium bicarbonate that helps to neutralize some of the stomach acid. And even though these are initially inactive, the enzymes are activated by enzymes in the small intestine. We'll come up to that in a second. So you've done a study on the anatomy of the pancreas and the liver, I think, in fair depth, at least the surface organization. And you know the common bile duct and the pancreatic duct combine in most people. You can't see it. The circle disappeared. But you know they combine in most people. In rare instances, and in the diagram in your lab from 2 weeks ago, it's shown as two separate ducts. And that's possible. But in the majority of individuals, the two combine, the common bile duct and the pancreatic duct combine into the hepatico-pancreatic ampulla. Ampulla? Ampulla. Call it ampulla. I think it's also a throwback to French because it's kind of bulb-shaped. Looks like a light bulb. Now, regulation of the exit of the juice from the pancreatic duct, from the hepatopancreatic duct, is from the hepatopancreatic, I keep saying that wrong, sphincter, or more easily, the sphincter of Oddi, which regulates release of bile and pancreatic juice into the duodenum when needed. So in the presence of food, that juice is released in order to help break down that foodstuff. And only after it's been released are the enzymes activated. And this is what protects the pancreas from itself being digested. That all of these enzymes, which are broad spectrum enzymes that work on all sorts of different bonds in different kinds of food, that they start inactive is what prevents the cells in the pancreatic duct and the pancreatic cells proper from being digested. These are activated by cells in the brush border of the small intestine as they travel through it. And we'll get to that shortly. And clearly it's supposed to be chyme with a Y, not chyme with an I, or chyme in this case. I'll have to thank autocorrect for that one. So one last note, we have this alkaline fluid with many enzymes being released to help counter the acidic chyme in the duodenum and help to process some of the food stuff. The common hepatic duct, of course, releases bile, which is the emulsifier that suspends fat in solution. Now you'll know from experience when you have a bottle of oil and vinegar salad dressing that it separates in two distinct layers. One's oil, one's water-based or vinegar-based, and they don't like to mix. And when you put that salad dressing on your salad, you don't just invert it and pour like a psychopath, you shake it up first. And by shaking it up, all those individual fat droplets are dispersed through the dressing. It looks uniform. That's the process of emulsification. Not one large layer separated, many individual small droplets that are surrounded by the juice. In this case, those small droplets are fat in the incoming food. And I'll bring this back up when we come back, but bile emulsifies and puts those fat particles in suspension. Small fat particles in suspension so enzymes can act on them. We'll come back to this on Thursday. Happy studying. Member exam is on Friday afternoon.